Да, уважаемые коллеги, я думаю, мы можем начинать нашу презентацию. Позвольте представить, сегодня мы проведем презентацию, озаглавленную «Культивирование плюрипотентных стволовых клеток человека без использования клеток фидеров». Презентацию любезно согласился представить доцент, доктор Масато Накагава из Центра по исследованию и применению индуцированных плюрипотентных стволовых клеток в Киотском университете. Я передаю ему слово, презентация будет на английском языке. Как я уже сказал, вы можете задавать ваши вопросы в чате на русском или на английском. Я переведу их для доктора Накагавы, и после презентации мы ответим на все ваши вопросы. Буду делать перевод на русский язык. Dr. Nakagawa, I just introduced your webinar, and if you can see the first slide of your webinar, we are ready to begin. Please start your presentation.
Okay, so thank you for a kind introduction. My name is Masato Nakagawa from Center for IPX Research and Application at Kyoto University. Today, so I would like to talk about the feeder-free culture system for human pluripotent stem cells. Okay, next slide, please. Okay, so this slide illustrates the potential application of iPSC technology. As you know well, so iPS cells can be generated from somatic cells of our body. And so once we obtain the iPS cells, we can produce several kinds of somatic cells in a culture dish. And using this iPS technology, there are several applications. So one is cell transplantation therapy, and the other is disease modeling. If the original cells has a disease-related genetic abnormalities, then the iPS cells have the same genetic abnormality. Using this disease model iPS cells, we can reproduce the disease in a cell culture dish. So using this disease model iPS cells, so we can do drug screening or toxicity test or side effect test. So this is the application of the iPS cell technology. So next slide, please. So today's topic is cell culture system for iPS cell or ES cell. So this slide illustrates the generation of the human iPS cells. So we can separate two steps. So the first step is establishment of iPS cells, and the later step is the maintenance of iPS cells. First, we need to transduce reprogramming factors into the somatic cells. 20 or 30 days after transduction of reprogramming factors, so we can see the human iPSC-like colonies in a cell culture dish. And after that, so we clone these iPS cells, and finally, we establish the mature human iPS cells. So next slide, please. So very recently, we studied about the one of the reprogramming factors, MYC. So MYC gene is well known as the oncogene, and so the first reprogramming factor includes the CMYC genes. So using CMYC genes, we can generate the iPS cells. So we found more effective factor that is named MYC-L genes. So CMYC and MYC-L are the same MYC family genes, but using MYC-L genes, we can increase the reprogramming efficiency in human cells. So now we usually use the reprogramming factor set, includes nickel. So this set is also used for the manufacturing of clinical grade iPSCs in Japan. Next slide, please. So we talk about the culture system of the human iPS cells and so human pluripotent stem cell. Next slide, please. So this is a photo of iPS cells. The area in yellow is iPS cell colony. There are thousands to tens of thousands of iPS cells in this colony. And so you will find that there are other cells around the iPS cell colony, fibro-like cells. So these cells are feeder cells. Feeder cells support the growth of iPS cells. So this co-culture system is called on-feeder method. So this culture method has been developed for the cultivation of the human ES cells at first. And so this culture method is very stable and so has a proven track record in research. So this on-feeder method is a good culture system for human iPS cells and so human ES cells. However, there are several issues with this culture method when considering its clinical application. So next slide, please. So there are several issues, so mainly two issues. So one is related to the medium and the other is related to the feeder cells. So between two issues, a common issue is the presence of the animal-derived components. So medium includes FBS, fetal bovine serum, also feeder cells are derived from mice. So both materials containing the animal-derived components. So using these animal-derived components, so there are several risks for clinical application. So when thinking about the future application, we reach the conclusion that we need to use a medium that does not contain animal-derived components and a culture method that does not use feeder cells. So next slide, please. So this slide shows the brief method of the on-feeder method for human pluripotent stem cells. Human pluripotent stem cells are co-cultured with feeder cells, green fibroblastic-like cells. And before the passage procedures, first we need to remove the feeder cells. And next, the pluripotent stem cell colony is broken into the small cell clumps, not single cell. So then small cell clumps are plated to the new cell culture dish with the fresh feeder cells. And the iPS cell or ES cells grow well with the feeder cells. So this is an on-feeder method. So this is very stable method, but there are some technically difficult points, such as making the small cell clumps. So we can not make same size of the small cell clumps at every time. So this is a very difficult point. And so previous slide I said, so there are some issues to overcome the clinical application. So next slide, please. So we have started to develop a new culture method as a feeder-free culture method. So to achieve the development of this feeder-free method, next slide, please. We needed to develop novel coating materials instead of the feeder cells. And so we needed to develop animal-derived component-free medium for future application. Next slide, please. So at first, I would like to explain the development of the coating materials. Next slide, please. So this is an image of the feeder cell culture system. So cell dish is coated with green colored coating materials such as recombinant protein or some polymers. And then iPS cells or ES cells are plated on these coating materials. So this is an image of the feeder-free culture system. So before development of this study, we have to understand how to bind the iPS cells or ES cell to the extracellular matrix. Next slide, please. So European research group has reported the mechanism of the binding of the iPS cells or ES cell to the extracellular matrix. So human pluripotent stem cells are expressing the integrin alpha 6 beta 1. This is the adhesion molecule on the cell surface. And so this integrin alpha 6 beta 1 can bind to the laminin type 511. So this is the extracellular matrix. So this integrin and laminin binding is very strong. And also using the recombinant laminin 511 proteins, so they could maintain well the human pluripotent stem cell. So we decided to confirm the function of this laminin 511. Next slide, please. So this is a structure of the intact full-length laminin 511. Laminin 511 is composed with three polypeptide chains, alpha, beta, and gamma chains. So as you can see, so this laminin 511 is very large molecular weight, so about 800 kilodalton. So also recombinant protein can be purified, but the purity was very low. So we thought this recombinant protein is not good for our purpose. That's future clinical application. So as you can see, so pink colored region is the integrin binding region in the laminin 511 proteins. Next slide, please. So in Japan, also university, Professor Sakiguchi has found that this integrin binding region, only this small portion of the laminin can bind to the integrin very efficiently. So this active laminin fragment is called as a laminin 511 E8 fragment. So Dr. Sakiguchi's group has already developed the production and purification method of this active E8 fragment. So molecular weight is smaller than intact laminin 511. And so this E8 fragment can be purified by the affinity column method. So result in the very high purity. So so next slide, please. So this E8 fragment is sold as I matrix 511 in the world. Next slide, please. So we found that laminin 511 E8 fragment, imatoric 511 is very good coating material for human iPS cell and ES cell. So next step to be resolved is medium. Next slide, please. So today I don't show any detailed data for development of this medium, but so we studied to collaboration with Ajinomoto company. So and finally we succeed to develop novel medium suitable for iPS cell named StemFit. So this medium is co-developed with Ajinomoto company and Kyoto University, and this StemFit does not contain animal-derived components, and is optimized for human iPS culture, also human ES cell culture. And so this StemFit shows a maximum effect in the cell culture by combination with imatoric 511 coating materials. So next slide, please. So we could find and develop a novel medium and coating materials for human iPS cell and ES cell culture system. And so we achieved to develop a feeder-free culture system of human iPS cells using this StemFit and so imatrix 511. Next slide, please. This slide, this is the first method of the imatrix coating in feeder-free culture system. So at first, we need to coat the cell culture dish with imatrix for over 1 hour before cell passage procedures. So after coating, so we dissociated the iPS colony into single cells, and the single cell plated in the imatrix coated cell culture dish. So this is the first imatrix coating method in the feeder-free culture system called pre-coating method. So this is a normal method. And so we use the amount of imatrix 511 for this pre-coating, 0.5 microgram per square centimeter. So very recently, another group in Kyoto University has reported the novel coating culture method. Next slide, please. The method is called mix method. So under this mix method, we don't need to pre-coat the cell culture dish with imatrix before the cell passage procedures. Only after the dissociation into the single cells. And so when we plate the single cells with imatrix, only this mixture can be plated to the cell culture dish. So using this mix method, we don't need to pre-coat the cell culture dish with imatrix. For this is the time-consuming procedures. And so even better thing is we can reduce the amount of imatrix 511 for this mix method in half. So the amount is 0.25 microgram per square centimeter. So imatrix is a little bit expensive, so material. So this mix method is good for our laboratory. So we use this mix method as our usual method for iPS cell culture. Next slide, please. So I summarize feeder-free culture method and on-feeder cell culture method. So for feeder-free culture method, we use the iMatrix 511 for coating materials and StemFit for medium. And so we can passage the iPS cells as a single cells under the feeder-free culture system. So then we can count the number of the single iPS cells and plate the exact number of the iPS cells in any time and anywhere. So split rate is very high compared to the on-feeder method. So under feeder-free method, we can passage one conflate dish to the 100 dishes. So we can easily expand the human iPS cell under feeder-free culture method. And so medium change is required every 3 days. So we can perform the weekend-free culture system. And so finally, we can make a freezing stock of iPS cells using the stem cell bank reagent. So at minus 80 degrees Celsius. So this is good and easy method for the making the freezing stock under the feeder-free culture system. So next slide, please. Could you show the movie? Thank you. So this is a time-lapse image. So at first, you can see the single iPS cells indicated as arrowheads. So please start. And so this single iPS cells can continue to grow, and so to make the large iPS colony. Please see this movie. So the single cells continuously grows, and so to form big iPS colony like this. Thank you very much. Please move to the presentation file. Next slide, please. Thank you. So I explained the good points for the feeder-free culture method. Next slide, please. So iMatrix 511 has been developed by the Dr. Sekiguchi Group, Osaka University, and NIP. NIP is a Japanese company. Next slide, please. And the StemFit medium has been developed by the Ajinomoto company and our group, Kyoto University. Next slide, please. And so these two materials, so PMDA consent has been acquired for clinical use. So this PMDA is agency in Japan, and the PMDA reviews the clinical application in Japan. So using these two components, so we can use this component for clinical use safely. Next slide, please. And so using this novel feeder-free culture system with iMatrix and StemFit, in Japan, Kyoto University group is now manufacturing the clinical grade human ES cells, embryonic stem cells. So these two materials is very safely used for the clinical application with iPS cells and so ES cells. Next slide, please. So we succeeded to develop a novel feeder culture system for clinical application. So next, I would like to explain the establishment of iPS cell. Next slide, please. So as for the establishment of iPS cells, the important point is a transduction method of the deployment factor into the somatic cell. Next slide, please. So I will explain the detail of the establishment of iPS cell. First, we use the retrovirus for the transduction method of the deployment factor into the somatic cell. So this is a use for the first time establishment of the human iPS cell in 2007. And so this is the first generation of the transduction method. And the second generation method is episomal vectors. So these are the plasmid vectors. So using these episomal vectors, we generate iPS cells. And this episomal vector system is now used for the establishment of the clinical grade human iPS cells in our institute. But this method, using this method, the transduction efficiency is so very low. So we need to increase the efficiency. Next slide, please. So now we are trying to confirm the sendai virus and synthetic RNA for iPS cell generation. So both methods is already sold as a product named cytotune or stem RNA. So using this method, we try to generate iPS cell. Next slide, please. So transduction efficiency is so very high using both methods. And so sendai virus is so one of the virus. So we need to handle sendai virus in biosafety level 2. But for RNA, we can easily and safely use normal experimental culture room. And using this method, I try to generate iPS cell. Next slide, please. First, I confirm the synthetic RNA for iPS cell generation. Next slide, please. So this slide illustrates the method of the iPS cell generation using the RNA. At day 0, we plated the HDF human dermal fibroblasts with the iMatrix 511. Next slide, please. And so day 1, 2, 3, and 4, we perform the 4 repeated transfection. Next slide, please. So the details write it here. And so we during this transfection, we use the medium StemFit basic 03 without basic FGF, not containing the basic FGF medium. Next slide, please. And after 4 repeated transfection at day 5, we change the medium to the StemFit basic 03, not containing the basic FGF. Next slide, please. And day 7, we change the medium to the StemFit basic 03, containing basic FGF. So next slide, please. And so day 10 to 14, I check the establishment of the iPS cell by morphology or flow cytometry to check the expression of the TRA160. TRA160 is a marker for a human pluripotent stem cell. Next slide, please. So this slide shows the morphology of the lip-grammed HDF at day 5 and day 11. I used another medium, Nutristem. So this medium is recommended in the attached protocol of the RNA kit. And I use basic 03 plus basic FGF, and so right photo, I use the basic 03 without the basic FGF. At day 11, please see the below photo. Using the Nutristem and basic 03, we can find the IPS-like colony in a well, but not in the basic 03 plus basic FGF. Next slide, please. And so the colonies and the cells are stained with Tron60 antibody at day 11. This is a result of the Nutristem medium. So as you can see, the colony is stained with Tron60. You can see the green signals in the colonies. Next slide, please. This is basic 03 plus basic FGF. So we can not see any like colony, but so some cells are stained with Tron60. But so we thought these cells are not iPS cells. Next slide, please. So this is a result of the basic 03 without basic FGF. So as you can see the very beautiful iPS colony. The iPS colony stained with Tron60. So next slide, please. And so we collected these cells and analyze the expression percentage of Tron60 by flow cytometry. And so the results shown in the below. So as you can see, using a basic 03 without basic FGF, right result. So almost cells are about 90.8% of the cells are Tron60 positive. So almost cells are reprogrammed by the messenger RNA using basic 03 medium. Next slide, please. So we confirm the synthetic RNA can be used for the iPS establishment and very high efficiency. Next slide, please. Today, I don't show any data about the Sendai virus, but using Sendai virus, we could also establish the iPS cells very high efficiency. The efficiency is similar to the RNA, messenger RNA. Next slide, please. And I show the more detailed results. So when we use the fibroblasts as the original cells for iPS establishment, so we could use both Sendai virus and synthetic RNA. But so when we use PBMC, peripheral blood-derived mononuclear cells, so we could establish iPS cells using the Sendai virus, but not with synthetic RNA. So, so this is a problem for our iPS generation. So, but so now we are trying to generate the iPS cells from PBMC using RNA technology. So next please. So I'd like to summarize today's talk. So we succeeded to establish the novel feeder-free iPS cell culture system. So using the iMatrix 511 and the StemFit medium, so you can perform the cultivation of human iPS cells very easily, since as ordinary cultured cell. And so also you can generate the iPS cell using the RNA, synthetic RNA or Sendai virus combination with the iMatrix and the StemFit. So please try these materials and feeder culture system for human pluripotent stem cell. Next, please. So this is the last slide. The data presented today was obtained with help of many people. So I would like to take this opportunity to thank all collaborators. So thank you for your attention.
Thank you very much, Dr. Nakagawa, for your inspiring talk. I will now ask in Russian for questions from our participants. Уважаемые коллеги, я поблагодарил доктора Накагаву за его прекрасный доклад по культивированию плюрипотентных стволовых клеток человека без использования клеток-фидеров. Если у вас есть вопросы, которые вы хотите задать прямо сейчас, пожалуйста, пишите их в чате. Если вопросов не возникнет, то мы всем участникам разошлем презентацию, и вы можете задавать вопросы по электронной почте. Я сейчас напишу в чате электронную почту. Так, вопрос от Ярослава. Каким методом отбирают, делают селекцию клеток после проведения трансдукции при получении IPS? Так, я сейчас постараюсь перевести этот вопрос для доктора Накагава. Доктор Накагава, there is a question from the audience. What method is used to select cells after you perform transduction when you generate induced pluripotent stem cells?
Okay. After transduction of reprogramming factor, about 20 or 30 days, so we can see the iPS-like colonies. And so normal method is pick up the colony by the manual method using the pipette man. So, or another method is after forming a colony, we collected the all cells at once, and then plated a single cell in a well using the 96 well plate, and select iPS colony using this 96 well plate. So we can select the pluripotent stem cell using this method.
Thank you, Dr. Накагава. Коллеги, я пытаюсь перевести, насколько я смог уловить этот долгий ответ. Значит, насколько я понимаю, после того, как клетки трансдуцированы, можно отобрать, то есть клетки индуцированные плюрипотентные стволовые проявляют себя в виде отдельных колоний. Можно взять все клетки, не отбирая какие-то отдельные клетки, и высеять их 96-луночные планшеты, и таким образом отобрать единичные колонии. Okay. Next question, Dr. Nakagawa. Is it possible to introduce to a human the cells transduced inducible pluripotent stem cells that you generate by your method?
What kind of cells can be reprogrammed?
Да, извините, уточнение. Вот вы говорите вопрос от Юлии Жеребцовой. Возможно ли введение человека трансдуцированных клеток? Про какие клетки вы хотите спросить? Про какие именно клетки вы хотите спросить? Пока Юлия уточняет свой вопрос, доктор Накагава, once we wait for the clarification of the question, yes, just clarification. We talk about inducible pluripotent stem cells. Is it possible to give a human these inducible pluripotent stem cells that are prepared by your method?
I think so. Yeah, you can prepare the IP surgery then.
Да, ответ да, да, можно приготовить. Next question, Dr. Nakagawa. Can you characterize briefly what are the main differences between StemFit medium and Essential 8 medium?
Okay. Yes. Previously, I checked the Essential 8 and the StemFit with the iMatrix 511. So StemFit is better compared to the Essential 8 for iPS cell culture. So the growth rate and so morphology is also better using the StemFit compared to the Essential 8. And so about the establishment of iPS cells. So the efficiency is very low using the Essential 8 medium. So we conclude that StemFit is the best medium.
Да, коллеги, если я опять же уловил ответ доктора Накагавы, в его экспериментах по сравнению среды Essential 8 и StemFit было показано, что морфология клеток и рост клеток лучше происходит на среде StemFit. Dr. Nakagawa, and connected question, if someone grows cells in Essential 8 medium with VNT matrix, how to transfer cells to the StemFit medium and laminin matrix? How the transfer is performed?
Okay. So you can very easily transfer your culture system to the StemFit and iMatrix system. So if so, you dissociate into the single cells. So please plate the cells using the Essential 8 and the vitronectin, VMG. And so after plating, 24 hours after plating, so please change to the StemFit medium at first. And so please culture for a week to grow the iPS cell. And so at the second passage, so please use the iMatrix and StemFit for the passage system. So you can easily transfer the next culture system.
Да, насколько я понял, можно легко перевести клетки на среду StemFit. Значит, при пересеве вы сеете клетки, как вы раньше их сеяли, на среде Essential 8 одиночными колониями, и через 24 часа вы заменяете всю питательную среду на питательную среду StemFit. А после 7 дней недели в культуре вы уже пересаживаете клетки, колонии, которые растут, и уже используете среду StemFit после того, как вы пересадили клетки. Коллеги, пожалуйста, напишите, правильно я перевел ответы доктора Накагава, чтобы было понятно, прояснили ли мы ситуацию. Thank you, Dr. Nakagawa.
Yep.
Да, коллеги, если больше нету вопросов, то, пожалуйста, посмотрите в чате. Я написал адрес своей электронной почты, по которой мне можно задавать вопросы для доктора Накагавы, и я смогу запросить у него ответы, если будут нужны более детальные объяснения протоколов роста клеток на StemFit. Также я хотел сказать, что эта среда должна быть без... Yes, Dr. Nakagawa, a connected question to the question previous one. Do we need to use a StemFit medium without basic fibroblast growth factor or with a basic fibroblast growth factor?
For the maintenance of iPS cells. So please use a StemFit with basic FGF. So basic FGF is so necessary for maintenance of pluripotency of iPS cells. Please use basic
Да,
FGF.
это было в презентации. Насколько я понимаю, те данные, которые показал доктор Накагава, без использования основного фактора роста фибробластов, они используются при трансдукции клеток в индуцированные плюрипотентные. Но для поддержки индуцированных плюрипотентных стволовых клеток необходимо использование основного фактора роста фибробластов. Так что ответ, да, для перехода, для поддержания индуцированных плюрипотентных стволовых клеток следует использовать основной фактор роста фибробластов. И я еще хотел сказать, Dr. Nakagawa, our participant thanks you for answers to his questions. Я еще хотел сказать, что у нас сейчас в наличии, у нас у компании Helicom в наличии на складе тестовые образцы с питательной среды StemFit и тестовые образцы основного фактора роста фибробластов. Пожалуйста, напишите мне запрос с указанием вашей лаборатории, и если можно, буквально несколько слов о том проекте, который вы ведете, для того, чтобы мы выслали вам бесплатно тестовые образцы такой питательной среды, и вы могли попробовать ее в своих экспериментах. Dr. Nakagawa, there are many thanks from the audience that I read in our chat. I think we need to finish now. There are no more questions. If there will be questions, then I will receive questions if there will be questions in the future, and I hope you will be able to answer them in private. If I can ask you so, we will very much appreciate this. Thank you very much, and we wish you good luck with your work. And thank you for joining and giving this very interesting seminar for us today. Thank you,
very
Dr.
much
Nakagawa.
for kind help.
Да, спасибо большое, уважаемые коллеги, что присоединились к нам сегодня. Я попрошу администратора вебинара завершить наш вебинар. Вам будет доступна видеозапись вебинара. Большое спасибо и успехов в вашей работе. До свидания.